a molecule of glucose uh, with the 2 molecules of ADP and inorganic phosphate um, then NAD, 2 molecules of NAD. It yields 2 molecules of pyruvic acid, 2 molecules of ATP, and 2 molecules of NADH2. So this is the thing, this glycolytic pathway, AMP pathway, takes place it's found in all organisms, both aerobes and anaerobes. So this is found alternately in specific some microorganisms. Of course, a hexose monophosphate pathway or a pentose phosphate pathway. This is found in all organisms, in aerobes and anaerobic organisms, all kinds, because in this the pentose sugars that are produced, so that are actually involved in the biosynthesis of nucleic acids, and of course the other than pentose sugars, it also yields 7 carbon sugar and 4 carbon sugar, erythrose sugars. And these particular things, this is seen in microorganisms which are grown, on pentose sugar source, source of carbon, pentose sugars. In those organisms, they make use of this, pathway, HMP or pentose PP pathway, to produce, 6 carbon sugar from 5 carbon sugar. And next thing is ED pathway. Of course, the Embden-Meyerhof pathway that I have shown. The net yield is 2 molecules of pyruvic acid, 2 ATPs. So 2 ATPs consumed and 4 ATPs produced, but the net production is 2 ATPs and 2 NADH2. Then hexose monophosphate pathway or pentose phosphate pathway, one and the same. In this case, of course, this is seen in both aerobic and anaerobic organisms. And here, the beauty of this things, when glucose undergoes this pathway, this thing, results, the 5 carbons, 4 carbon, 5 carbon, pentoses or erythroses, 4 carbon sugars, or sedaheptaloses, 7 carbon sugars, and hexoses, and 3 carbons, trioses. All these things are produced, and of course, among them the 5 carbon sugars which are produced mainly in the form of ribulose, ribose. They are made use in the production of in the biosynthesis of nucleic acids. And of course, the other thing information is that in case of when the microorganisms are grown on a 5 carbon sugar, source of 5 carbon sugar, of course 5 carbon directly can't enter the the catabolic pathway, so it has to go back and comes back from 6 carbon state. So from when they are grows on pentose phosphate, pentose sugars, then by using this pathway, these organisms, they produce 6 carbon sugars to make use a source of energy. So that's the things, and the net production, of course, pyruvic acid, NADH and the ATP, they are produced. The next thing is Entner-Dudroff pathway. Entner-Dudroff pathway, it is seen, in Pseudomonas organism, and in this particular, the key compound that is produced in the organisms which follow this, a key compound that is KDPG, that is 2 keto 3 deoxy 6 phosphogluconate. It is found in page number 31. And of course, in this pathway, like both NADH and NADPH both are produced along with the ATP. along with the ATP. Then phosphoketolase pathway. Phosphoketolase pathway, in this particular things, like 5-carbon intermediate is also produced, and the key compound is phosphogluconate. And of course, all these pathways alternate EMP pathways, like you have Hexose monophosphate pathway or phosphoketolase pathway or Entner-Dewdorf pathways. The organisms which follow these pathways, which possess these pathways, so they of course like make use of this pathway along with the EMP pathway. At some part of the pathway, actually the intermediaries, they are passed through EMP pathway to produce pyruvic acid and NADH and ATP. So and of course, after this, we have also actually we had gone through this particular things, the information on fermentation, which is a type of metabolism where both electron donors and electron acceptors are organic in nature. And another important aspect of fermentation is that the electron acceptors are endergonic, produced endogenously. That thing, whereas in respiration, the electron acceptors, they are external. They come from externally from external source. They are made use, either oxygen in case of aerobic respiration, sulfate or nitrate, or other things in case of anaerobic respiration. But in fermentation, these electron acceptors both are produced endogenously. So, and and another thing is oxygen is not required for these things, and in this case, no additional ATP is generated, by following things. And only thing is, in this, ferment the organisms which follow fermentative pathway, many, of course, like either alcohol or organic acids, different kinds of organic acids are the end products of the fermentative organisms. And of course, the main important things, though this pathway doesn't generate much energy, but it, these things are the end products, are the intermediates of the fermentative pathway, or made use of the indicators for biochemical identification. And also majority of them, so they have the importance, economic importance. For example, here, lactic acid fermentation, where the homofermentative lactic acid bacteria, like the different species of lactobacillus genus, or the streptococcus, species of streptococcus are involved. So the industrial production of lactic acid. Then mixed acid fermentation, that is Escherichia coli. So this is actually very important. So this particular thing is used in the identification of working on coliform organisms. That is, you must be aware in the thing, like like drinking water, its purity, it is judged by a test called potability test. So the the fecal contamination things, where E. coli is used as an indicator organism. So here we make use of a test called methyl red and Vox-Prosk reaction. So MR test, that particular thing, it answers. And similarly, the heterofermentative lactic acid bacteria, represented by Leuconostoc, and other lactic acid bacteria genera, they produce a set of organic acids in addition to lactic acid. Of course, they too have the economic importance. So both homofermentative and heterofermentative lactic acid bacteria, they are made use in the preparation of various dairy products, various fermented vegetable products, then fermented sausages and so on. Then the other thing is, the 2,3-butane diol fermentation, which is seen in a coliform bacterium, Enterobacter aerogenes. Of course, this is again used, made use in a test called Wilks-Proska reaction. That, of course, in your practical things, you will come to know about them. So their importance. So this is thing, the reaction where the lactic acid is produced from the end product of glycolytic pathway, pyruvate or pyruvic acid. So how it is produced. Then in the process, the NADH that is formed in ethanol producing organisms from the pyruvate which produce ethanol. Of course, this NADH, it is again reoxidized to NAD to facilitate the continuation of the pathway. Of course, they mixed acid fermentation. In this case, varieties of organic acids that are produced in addition to lactic acid, like other acids like acetic acid, formic acid, even carbon dioxide, carbonic acid, even hydrogen gas, all these things are produced. This is, of course, in the yesterday's class, I told you, in hot summer sometime, the curd loses its natural aroma. So the, it, it will get the flavor or aroma of the toddy, or some other things, and things, that is due to the production of the contamination with, the heterofermentative lactic acid bacteria, mixed acid fermentative organisms. Then butanediol fermentation. Of course, this is, of course, how it follows. Sort of from pyruvic acid, acetolactic acid is produced, then it is converted to AMC, a product called AMC, acetyl methyl carbinol or acetoin, and in turn, then it is converted to 2,3-butane diol. This particular compound presence is, made use in a test called MRVP test, methyl red. Then the second part of it, that is Wilks-Proska reaction, this is made use, and the organism, important example of the organism which follows this pathway, it is Enterobacter aerogenes. Then, of course, the importance of fermentation as a thing, as a metabolic type. Of course, it has many like economic importances, like it is made use in ethanol production, large scale. Then ethanol production, then propionic acid bacteria, which is used in a type of in the production of a cheese type, that is Swiss cheese, with characteristic eye buds, and with the bitter flavor, propionic acid, Propionibacterium and its species Shermani. It is used in the production of Swiss cheese. Then similarly, Clostridium mastobutylicum, which is used in the production of acetones, butyraldehyde, butanol, and all, which have the thing, the industrial importance, industrial solvents. And the down you have many examples of the thing, the fermentative organisms and their products, what they think, that is the things, Streptococcus, Lactobacillus, and even including Leuconostoc, like they are made use in the production of curd, then yogurt, then cheese, and so on, and in the production of varieties of fermented vegetables like sauerkraut, pickles, and fermented sausages, and so on. Then, of course, ethanol, it is you can have the examples like wine making, like beer fermentation, then the production of distilled alcoholic beverages, and so on. Butyric acid, the organism Clostridium acetobutylicum, which is used in the production of a set of compounds which have the industrial importance: butyric acid, butane, acetone, isopropanol, and so on. All these things. So then, of course, propionic acid, I have already told you, this is used in the preparation of a kind of cheese, that is Swiss cheese. Then mixed acids, which is, of course, this fermentation by E. coli. It is used in a biochemical test called methyl red test. Then 2,3-butanediol, which is followed by enterobacter aerogens, which is used in a test called Vogt-Proska reaction. That is, both of them together, they are used together. That is, together it is called as MRVP test, methyl red Vogt-Proska reaction. Then, of course, coming to the respiration, of course, the respiration, I have told you, of course, even in theory class and several occasions previously, the all living organisms, they possess two kinds of thing, the metabolism of these living organisms is broadly classified into two categories: the fermentation and respiration, based on the electron donors and electron acceptors. Mainly, what makes difference is, it is not the electron donor, it is the electron acceptor. In case of fermentation, both electron donors and electron acceptors, both are organic in nature, and the electron acceptor, it is one of the intermediates, reduced product. It acts like oxidized product. It acts like so the acceptor. So acceptor, electron acceptor, it is again organic, both the electron donor and electron acceptor. Whereas in respiration, the electron donors can be either organic or inorganic, but the electron acceptor has to be inorganic. Other point is, they are exergonic, means external. They are they are made use from the external source. So in case if it is oxygen, you call it as aerobic respiration. In case if it is other than oxygen, a substance usually inorganic other than oxygen, so like nitrate, nitrite, sulfate, and so on, even CO2 in methanogenic organisms, in that case you call it as anaerobic respiration. But whether it is aerobic or anaerobic respiration, the electron acceptors to be called as respiration has to be compulsorily inorganic. So then only they get the name respiration. Then of course, this is the thing of respiratory metabolism, and till pyruvic acid, glucose to pyruvic acid, all organisms follow that pathway. So both anaerobic and anaerobic organisms. And in anaerobic organisms, further they will not use the other cycle, TCA cycle or citric acid cycle. It is absent in these organisms. But instead, the end product of the glycolytic pathway, it is diverted to produce, depending on their metabolic makeup, either alcohol or different kinds of organic acids or acids and so on. So in these, organisms which follow fermentative pathways. So whereas in respiration, the end product of glycolytic pathway, it enters next cycle, that is the Krebs cycle. It's called as Krebs cycle or TCA cycle, tricarboxylic acid cycle, or citric acid cycle. So it is called as citric acid cycle mainly because the first product, the visible thing that is produced in this Krebs cycle, it is citric acid. This citric acid is produced from pyruvic acid. So by like combining a molecule of pyruvate along, a molecule of, of course, initially the pyruvate is converted to acetyl coenzyme A, And this acetyl coenzyme, pyruvate enters TCA cycle not directly, it enters through acetyl coenzyme A, but this acetyl coenzyme A, it combines with oxaloacetic acid. Oxaloacetic acid, so to produce, citric acid. The first visible thing, that is formed is citric acid. This is the, of course, before that, the conversion of pyruvate to acetyl CoA, it is the transitional step. It is called as, during this step, what happens? Pyruvate gets converted to, it loses, it, a molecule of, like things, acetyl CoA, it loses the decarboxylation, it undergoes, and to produce acetyl CoA along with oxaloacetic acid, which is an intermediate of the citric acid cycle, or a Krebs cycle or TCA cycle, to form citric acid. This citric acid, of course, it undergoes, it goes through many intermediates, because it is a cycle. It goes on forever, ever. So the citric acid becomes isocitric acid, then alpha ketoglutaric acid, then it becomes succinyl coenzyme A, then it becomes succinic acid, then formic acid, fumaric acid, succinic, fumaric acid, then malic acid, then oxaloacetic acid, again oxaloacetic acid, of course, because it, oxaloacetic acid, the importance in the metabolic pathway, they are interdependent. Like the end product of glycolytic pathway, it is important. Availability of pyruvate to form acetyl CoA, for the oxaloacetic acid to form citric acid. That's the thing, so it goes on, This TCA cycle, which is formed, it is, of course, it enters in the metabolic pathways of the living organisms. It occupies the central stage of metabolism. It is called as central stage. So TCA cycle or Krebs cycle or citric acid cycle, it occupies the central stage of the microbial metabolism, where you find the things, the branching out of the catabolic pathways and the anabolic pathways. So because the intermediates of the TCA cycle, they are made use of the raw materials for the biosynthesis of various things, amino acids, then nucleic acids and fatty acids and so on. They are diverted to produce these things, so especially oxaloacetic acid and such kind of things are produced. So they are made use for this particular thing. So since it is, of course, in addition to participating in the generation of energy, since TCA cycle or Krebs cycle, it also provides many important raw materials for the biosynthesis of cellular macromolecules, like carbohydrates, lipids, proteins, and nucleic acids. It is called, as, it is amphibolic cycle. So the TCA cycle completes oxidation of glucose to produce, and of course at the end of that, which are all the things, the resulted things in the, when glucose undergoes this TCA cycle, 2 molecules of carbon dioxide, 2 ATPs, 6 NADH, 2 FADH2, and of course here ATP means 1 ATP and 1 GTP. There are doesn't make, but it is taken as ATP only. Then various precursor metabolites for the biosynthetic pathways. This is the ultimate things of the Krebs cycle. Of course, Krebs cycle, while talking about Krebs cycle, you must remember it is many times it is referred to as amphibolic cycle. And another thing, it occupies the center stage of both catabolic and anabolic pathways, because one side it produces NADH2, FADH2 and all that, which further when they are passed through terminal electron, the transport system, yields ATP molecules. At the same time, the intermediates of the Krebs cycle, they are made use of raw materials for the biosynthesis. All these things are given in this particular things. So of course, the next important things, after Krebs cycle, the important component of the aerobic respiration, it is the respiratory metabolism. It is the electron transport chain. Electron transport chain. This is in prokaryotic organisms. So it takes place in the cell membrane, the bilayered cell membrane. It is this particular thing is found. Electron transport chain is found in the cell membrane of the prokaryotic organisms. So that bacteria. Whereas in eukaryotic organisms, where is it found? It's found in the inner mitochondrial membrane, the cristae. Inside the mitochondrial membrane, so the mitochondria in the eukaryotic organisms, this is found, and this is arranged in the form of three complexes: complex 1, complex 2, complex the complex 3 and complex 4, and at the end ATP synthase. And in the, of course, the arrangement of this particular thing will be able to see in this particular things the components of the electron transport chain. So how it is arranged: in the first stage, quinones are there; in the second stage, cytochromes are there, and the flavoproteins are part of this electron transport chain. So actually, the main role of this electron transport chain is for the thing, the generation of protonomotive force. That is a gradient. A gradient is created, molecular gradient is created, electron gradient, where the thing, like in the beginning, of course, the electron transport chain, it is in the highly reduced state. It is in highly reduced state, and as you move towards the towards the end of that particular thing, the things, the redox potential slowly increases. It is highly reduced in the beginning, and it is like oxidized, oxidized in the towards the end of this particular pathway. So here the complex 1, complex 2, complex 3, and complex 4, they are arranged to all these things lead to ATP synthase, which of course the place where the ATP molecules are synthesized. And of course in this pathway, some of the things that are involved are ubiquinones and cytochromes, and all these things are involved in the in the formation of the electron transport chain. And the electron transport chain, of course, is membrane embedded electron carriers. So they pass electrons sequentially, eject protons in the process. In the process, and in prokaryotes, where this is found, this electron transport chain, it is found in cytoplasmic membrane, whereas in eukaryotes, it is found in the inner mitochondrial membrane. That is the thing, like the cristae, the structures, like membranous structures, double membrane structures that are found in the inner part of the mitochondria. Then energy gradually released, and this release coupled to ejection of protons at the end. These protons, protons create electrochemical gradient. That's what that is you call the protonomotive force. You use the word protonomotive force, where in the inner side it is highly reduced, outer side, the thing, little oxidized. See, the redox potential, they have, it is reduced redox potential in the inner side in the beginning, and oxidized redox potential in the towards the end of the things. So the electron transport chain. And some of the molecules which are part of this electron transport chain are quinones, cytochromes, and flavoproteins. And quinones, which are found in the electron transport chain, and they are lipid soluble molecules, move freely and can transfer electrons between complexes. Then the cytochromes contain heme protein molecules with iron atom at center of, and there are several types, the cytochrome 2 and 3 are there. Then flavoproteins to which flavin is attached. So the FAD or other flavin synthesized from riboflavin. And here another question arises: when these NADH2 and or FADH2, when they are passed, of course, before we discuss that, we shall go through this particular thing, the electron transport chain, which is involved in generating protonomotive force. And here, so which is seen in eukaryotic organisms in the mitochondria, whereas in prokaryotes, it is seen in the cell membrane. So there are different complexes: complex 1, complex 2, complex 3, and complex 4. At the end, you have that synthase, ATP synthase. And complex 1, here, the complex 1, it accepts electrons from NADH. And here you must remember, you should know that the NADH2 that are produced, they yield 3 ATPs, whereas FADH2 that are produced in NADH2, when they are passed through, from TCA cycle, when they are passed through electron transport chain, it yields 2 ATPs. Why is that difference? The difference is that the NADH2 molecules, they enter this electron transport chain bit earlier than the FADH2 molecule. So the NADH, it joins the electron transport chain in complex 1 stage only. So the this complex 1 accepts electrons from NADH and transfers to ubiquinone. So that's and in the process, it pumps 4 protons. Then complex 2, succinyl dehydrogenase complex, it accepts electrons from TCA cycle via FADH2. So FADH2 comes in the second stage. So then the downstream of those carried by NADH2, and it transfers electrons to ubiquinone. So since the NADH2 enters one step earlier compared to FADH2, NADH2 yields 2 ATPs, FADH2 yields 3 ATPs. Then, of course, the complex 3 mainly consists of cytochrome B and C, accepts electrons from ubiquinone from complex 1 and or complex 2, 4 protons pumped, electrons transferred to cytochrome C, and ultimately complex 4, which involves cytochrome C oxidase complex, which actually cytochrome C, which is in reduced state by accepting electrons from the previous, complex 1 and complex 2. This in turn transfers electrons to get itself oxidized. So this cytochrome C oxidase complex accepts electrons from cytochrome C and pumps 2 protons. And at the end, you have terminal oxidoreductase, meaning it transfers electrons to terminal electron acceptor. So in case of aerobic organisms, it is the oxygenous, the terminal electron acceptor, whereas in anaerobic or respiratory organisms which follow anaerobic respiration, it is not oxygen, but oxygen containing molecules like nitrate, nitrite, sulfate, all these things are produced. This is again another pictorial depiction of the respiratory chain. Of course, this is given, of course, in the mitochondria in eukaryotic organisms. Inside you have the cristae, in that particular things, the double layered membranes. Across that membrane, so the gradient is created. Gradient that the chemical gradient is created for the thing, to facilitate the production of ATP molecules. And of course, this the things shows in anaerobic organisms, and aerobic and anaerobic respiratory organisms. So the details about the electron acceptors, different kinds of electron acceptors that are used in aerobic respiratory organisms. And here all eukaryotic mitochondria, that is, you have plants, animals, then you have among the microorganisms you have the fungi, the algae, and other organisms. So all these things, oxygen is used as, and of course many bacteria, aerobic bacteria, oxygen is used as terminal electron acceptor. Then anaerobic respiration, the organisms which follow anaerobic respiration. Here, this is of course nitrate. Nitrate is used as terminal electron acceptor in nitrifying bacteria. The organisms which are involved in denitrification, some facultative microorganisms. Like for example, in several gram-negative enteric bacteria also, like you have Escherichia coli and Enterobacter and several kinds of that, enteric bacteria, gram-negative bacteria which colonize colon region of the intestine, plus many bacteria which are involved in nitrate reduction. So they use nitrate as terminal electron acceptor. Nitrite reduction. So this is of course both the Pseudomonas, Bacillus, and many organisms that are together, nitrate or nitrite. They are part of a process called denitrification, where initially nitrate is used as terminal electron acceptor to get itself converted to nitrite. Then further it is converted into nitric oxide, then to nitrous oxide, and ultimately to nitrogen gas. So this is actually these electron acceptors used in nitrate reduction. Then the next thing is in case of if sulfur is used. Sulfur is used as sulfate reducing organisms, disulfur vibrio and disulfur to maculum. So the sulfate is used as terminal electron acceptor to cause reduction of sulfur. In some organisms like disulfuromonas, elemental sulfur is used as a terminal electron acceptor to get reduce it to hydrogen sulfide. So of course in this it is shown at the end of the aerobic respiration in prokaryotic microorganisms, the ATP generation at different points. For example, in the beginning, in this it is shown the glycolytic pathway, where glucose is oxidized to pyruvic acid, oxidizes glucose to pyruvic acid. And at this stage, how many ATPs and how many NADH are produced, all that it is shown. Then of course the end products of glycolytic pathway are two molecules of pyruvic acid. Each molecule via acetyl-CoA enters TCA cycle, and when some of the intermediates of TCA cycle, in TCA cycle ultimately 6 NADH and 2 FADH2 are produced. In turn they give rise to, of course, from NADH2, each molecule of NADH2 you will get 3 ATPs, and each molecule of FADH2 you get 2 ATPs. And from the totally from all NADH2 from glycolytic and pathway and TCA cycle, how many ATPs are produced, and all that it is given at the end. So In aerobic respiration, you'll get, of course, 36 plus 2, that is 38 ATP molecules. So that's all that it is given here. Aerobic comparative view of fermentation and the respiration. In case if an organism follows aerobic respiration, what is the yield of the ATP? It is about 36 to 38 ATPs are produced. Whereas in anaerobic respiration, in the organisms which use alternate electron acceptors like sulfate, nitrate, carbonates or carbon dioxide and so on. So what is the yield? It ranges from 2 to 36 ATPs. And whereas the organisms which follow fermentation, like those alcohol producers, organic acid producers, and all these things, where the end product of them, either it is either alcohols or organic acids, then only they realize only 2 ATPs. But you must remember that the organisms which follow the fermentative pathway, they can, they have more, they can survive better because the terminal electron acceptors, they need not depend on outside source. So that is the electron acceptor, it is one of the organic intermediates of their metabolism, which is used. Thus they can survive better, though they, of course, they get, like they derive less energy compared to very less, minimum energy compared to the organisms which follow aerobic respiration. That's either anaerobic or aerobic organisms. Of course, the one, aerobic organisms, they derive more energy, up to 38 ATP molecules. So this is about the catabolic pathway, the organisms that follow. So in the catabolic pathway, catabolism. So what happens when a molecule of glucose, and you must remember in the beginning I have told you, this is explained with respect to carbohydrates. Carbohydrates, not all, the building blocks of the carbohydrates, simple sugars, in that the main hexose, sugars, 6 carbon sugars are able to, participate in the catabolic pathway. That's why the other, forms of sugars like, 4 carbon, mainly 5 carbon or other forms of sugars. So they are converted initially to 6 carbon state, and then they are actually, so they'll, this 6 carbon sugar is made to enter the catabolic pathways. Then the next thing, this is of course all that we have explained, like discussed so far. It is in heterotrophic organisms or organotrophic organisms, organotrophs. And then, of course, in among the microorganisms, you have autotrophs and heterotrophs. So in heterotrophs, of course, how it happens, the preformed organic molecules serve both as source of energy and carbon. That I have told you. In the practical class, in the nutrition category, in the preparation of media, we have discussed about this. Whereas the other group is autotrophs, the organisms which are able to synthesize their cellular macromolecules by using simple inorganic materials. So by definition, they are those organisms which use carbon dioxide as source of carbon. Means they have a mechanism in which they will be able to fix atmospheric carbon, that is carbon dioxide. They will be able to incorporate it into metabolic organic intermediates, that is ribulose, like 5 carbon sugar. They'll be able to do that. Whereas in both the in autotrophs, but depending on the energy, whether they derive energy from reduced inorganic molecules or from sunlight, they are further classified into chemoautotrophs or photoautotrophs. Chemoautotrophs, otherwise also called as chemolithotrophs. Lithotrophy means lithos means inorganic, elemental. So it is synonymously used. Chemoautotrophs or chemolithotrophs, one and the same. Photoautotrophs or photolithotrophs, one and the same. So the first, in case of coming to autotrophs, the first we shall consider the chemolithotrophs. They are those, of course, chemolithotrophs. They represent only prokaryotic organisms. They are, only in bacteria you see this kind of organisms. They are a unique prokaryotic organisms which have the ability to use reduced inorganic compounds as source of energy, like hydrogen sulfide in sulfur oxidizing bacteria, ammonia in case of nitrifying bacteria. Examples, two examples like that. You have several other examples, like even iron oxidizing bacteria. Iron oxidizing bacteria also key examples for this chemolithotrophs or chemoautotrophs. And this hydrogen sulfide or ammonia, which are actually which are produced in anaerobic respiration. For example, hydrogen sulfide in anaerobic respiration, for example, when sulfate is used as terminal electron acceptor, or when elemental sulfur is used as a terminal electron acceptor, it results in the formation of hydrogen sulfide, the reduced sulfur compound. Or when nitrate or nitrite are used as terminal electron acceptor, it results in the production of ammonia, a reduced nitrogen compound. So here in this statement, it is same thing. It is given here. The either hydrogen sulfide or ammonia, which are produced in anaerobic respiration from inorganic molecules such as sulfate, nitrate, and etc. They are serving as terminal electron acceptors. Actually, they are actually made use by these organisms for generation of energy. And the examples, different kinds are given here. For example, there are different types of these chemolithotrophs: hydrogen bacteria, sulfur bacteria, iron bacteria, nitrifying bacteria. 4 classic examples are given. Hydrogen oxidizing bacteria, what these they oxidize hydrogen to form water molecule, and hydrogenomonas is an example. And the next is sulfur oxidizing bacteria. Of course, in this both non-photosynthetic and photosynthetic both categories are there. When we but in the non-photosynthetic photosynthetic types, it is taken later in the photosynthetic organisms. Here, this particular class, because they are chemolithotrophs, sulfur oxidizing bacteria, that is non-photosynthetic sulfur oxidizing bacteria. They use hydrogen sulfide as source of reductant, and where this hydrogen sulfide is oxidized to either elemental sulfur, or in turn from elemental sulfur to sulfate they are produced. And the examples like the thiobacillus, acidothioBacillus, Beggiotova, Thiothrix, they are some of the examples. Finally, you have oxidizing bacteria, iron oxidizing bacteria. Here what happens, I have told you in, I think, the in media preparation or some practical class I have explained the elements, some of the polyvalent elements you call. The elements are found in different stages of oxidation. For example, iron, if you take iron, ferrous and ferric, and in this case the ferrous is reduced state. The reduced iron, ferrous, is by these organisms, it is oxidized to form ferric. In the process they derive energy. And the examples, of course, Gallionella, Spirotillus, and so on, different ferrobacillus, all these things are examples of iron oxidizing bacteria. They are also called as iron oxidizing sheathed bacteria or stalked bacteria. Both forms are types or examples for this category. Next category is nitrifying bacteria. Nitrifying bacteria. This nitrifying bacteria, of course, they use ammonium, which is produced, during anaerobic respiration, or which is produced during ammonification process. So it is utilized by these organisms, and the oxidation of ammonium to nitrate happens in two stages. Initially, from ammonium to nitrite by nitrosomonas and related organisms. In the second step, nitrite to nitrate by nitrobacter and related organisms. And the next is photosynthetic organisms. They are also called as photolithotrophs or photoautotrophs, and mainly represented by, of course, the plants, the cyanobacteria, that is oxygenic photosynthetic bacteria and anoxygenic photosynthetic bacteria. Two categories I have told you the examples. Oxygenic photosynthetic bacteria, they are the photosynthetic bacteria which have the oxygen evolving photosynthesis similar to higher plants, whereas anoxygenic photosynthetic bacteria, which is represented by two major groups of bacteria, that is green sulfur bacteria and purple sulfur bacteria. So both categories, and of course the primary pigments in these oxygenic photosynthetic bacteria, it is chlorophyll A, whereas anoxygenic photosynthetic bacteria represented by the chlorobium and chromacium. Chlorobium, it is green sulfur bacteria. Chromacium, it is purple sulfur bacteria. They have bacteriochlorophylls. Whereas which you have in the photophosphorylation two cycles, photosystem 1 and photosystem 2, cyclic and non-cyclic, and the oxygenic photosynthetic bacteria, they follow both photosystem 1 and photosystem 2, whereas the green sulfur bacteria follows only photosystem 1, whereas the purple sulfur bacteria follows photosystem 2. Of course, with respect to things, like in oxygenic photosynthetic bacteria, it is non-cyclic photophosphorylation, whereas in the green sulfur bacteria, non-cyclic photosystem, whereas in purple sulfur bacteria, it is reversed electron transport mechanism is used for that purpose. Then the source of electrons, of course, in oxygenic photosynthetic bacteria, it is water molecule, because photolysis of water results in the evolution of oxygen, whereas in the sulfur bacteria, anoxygenic photosynthetic bacteria, it is various reduced sulfur compounds like H2S, even sometime H2 is also, or some organic compounds are also used. Then the carbon fixation, by which how this carbon is fixed, because this photosynthetic bacteria, they are also carbon fixing, so they use, of course, Calvin cycle in both oxygenic photosynthetic bacteria, purple sulfur bacteria Calvin cycle, and green sulfur bacteria reverse TCA cycle. So the accessory pigments in these organisms, of course, the higher algae and the cyanobacteria, they possess both phycobiliproteins and carotenes in them, whereas purple and green sulfur bacteria, they do possess both of them. Of course, the carbon fixation, in both chemolithotrophs and chemoautotrophs, they fix carbon. The one, the classic carbon fixing pathway, it is Calvin cycle, where there are in each cycle of this Calvin cycle, a molecule of carbon dioxide is fused with an intermediate 5-carbon intermediate of this cycle, that is ribulose biphosphate. Ribulose biphosphate, ribulose, that is one ribulose 1,5 by biphosphate, that is 1,5, two phosphate molecules are it is called as, that is keto sugar, it is ribulose, right? Not ribose, ribulose, aldose sugars, it is keto sugar. So ribulose biphosphate carboxylase. To this ribulose biphosphate, one carbon is added to make it a 6-carbon state, but 6-carbon state is not produced to produce 2 molecules of 3-carbon compounds, 2 phosphoglyceric acids. Glyceric acid, 2 molecules of phosphoglyceric acid are formed in this particular stage. So, and of course they consume a lot of energy, 18 ATPs and 12 NADH, DPH, and are used by these organisms. Then anabolic pathways, of course, so catabolism and anabolism. Anabolic pathways, of course, the classic anabolic pathways are lipid biosynthesis, nucleic acid biosynthesis, and even the amino acid biosynthesis. These are all the 3 classic examples. Of course, rarely in some organisms, through gluconeogenesis, they think from the intermediates of the TCA cycle, the carbohydrates are deposited in the grains. Of course, in the plant products, like for example pulses, it is the protein, whereas in cereals it is starch. In some fruits it is the sugars, fructose or other forms of sugars. And then in oil, oil seeds, for example castor seed, then the peanut, this groundnut, and many things, for example even in sunflower, all these things, the reserved food material is lipid. And like how these are, the plants are able to accumulate respective type of these polymers, organic polymers, it is through this way, by following anabolic pathways. So like by like diverting one of the intermediates of the TCA cycle to produce the fatty acids in lipid biosynthesis, or purines and pyrimidines in nucleotide synthesis, or in amino, different amino acids in the protein synthesis, or gluconeogenesis, that is also formed in the thing, carbohydrate things, like biosynthesis. Of course, kindly go through this carefully. The anabolic pathways, how it is happens in a different kinds, in organisms. Of course, the from the glycolytic pathway and TCA cycle, from these respective catabolic pathways, how the intermediates are taken out and used in the production of either amino acids or fatty acids in lipid biosynthesis, or lipid biosynthesis. All that it's given in this particular things, fatty acids, amino acids, in then nucleic acids, nucleic acids, that's mainly nucleotide, like here it is glycolytic pathway, PPP, HMP pathway, or PPP pathway. Of course, the next thing is, let us see, let us conclude our time now. 14. Of course, this is of less importance. The nitrogen cycle, of course, where so this we will discuss in the next class. We will stop the class at this stage. Okay.
Yes, sir.
We will continue of course, when the little is there, and the next class, we will take up, let us say, whether microbial genetics or some part we will discuss, and practical, of course, which is the next aspect to be considered, is biochemical reactions of microorganisms. Of course, we in the previous things, we have dealt what is that, isolation, purification, and preservation of microorganisms, followed by the staining techniques in microbiology. Staining techniques, different kinds of stains and staining techniques that are followed. The next thing is biochemical reactions. So the different key reactions of carbohydrate or protein or lipid or nucleic acid metabolisms, which are made use in identification of microorganisms. So that particular thing, it is there, they are very interesting things, uh, which are actually in practicals we used to do with the thing, some colored compounds that are produced, they are very interesting things that to conduct the practicals. Unfortunately, this time you are not fortunate enough to have the practicals of them. But the only thing, at least we will follow the theoretical aspects with the protocols, which you can write down the records and preserve them for future use. So thank you all.
Thank you, sir. Thank you, sir.
Thank you. Thank you, sir.
What shall we do? Practicals. What shall we do?
Sir, I'll inform you, sir.
ಅದು ಹೇಳಿ, ಇದಿದ್ರೆ
ಹಾ,
ಅದು
sir.
ಮಾಡೋಣ. ಈಗ ಈ ವೀಕು ಬಯೋಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ಸ್ ಬರ್ತವ,
ಹಾ, sir.
ಮಾಡೋಣ. ನೋಡಿ, ನಿಮಗೆ ಟೈಮ್ ಇದ್ರೆ ಇದು ಮಾಡಿ.
ಆಯ್ತು, ಸರ್.
ನೋಡಿ, ಇದ್ರೆ ಶುಕ್ರವಾರನ ಯಾವಾಗ ಸಿಗುತ್ತಾ ಅಥವಾ ಎಲ್ಲರೂ ಮಾಡ್ತಿದಾರ?
ಸರ್, ಎಲ್ಲರೂ ಮಾಡ್ತಿದ್ದಾರೆ ಸರ್. ಅಂದ್ರೆ ಮಿಡ್ ಟರ್ಮ್ ಏನೋ ಮಾಡ್ತೀವಿ ಅಂತ ಅಂದ್ರಂತೆ ಸರ್, ಹಂಗಾಗಿ ಎಲ್ಲಾ ಪೋರ್ಷನ್ ಕಂಪ್ಲೀಟ್ ಮಾಡ್ತಿದ್ದಾರೆ ಸರ್.
ಒಮ್ಮೆ ಅಲ್ಲ, ಏನ್ ಮಾಡ್ದ ಅದ್ ನೋಡೋಣ, ನಾಳೆ ಕೇಳ್ತೀನಿ ಅವ್ರಿಗೆ.
ಹಾ, ಸರ್.
ಯೂನಿವರ್ಸಿಟಿ ಅದೇ ಡೀನ್ ಆಫೀಸಿಗೆ ಫೋನ್ ಮಾಡಿ ಕೇಳ್ತೀನಿ ವೆದರ್ ಅದು ಏನಾರು ಇದೇನಾರು ಲಾಕ್ ಡೌನ್ ಇನ್ನೂ ಎಕ್ಸ್ಟೆಂಡ್ ಆಗುತ್ತೇನೋ ನೋಡ್ಬೇಕು. ಯಾಕಂದ್ರೆ ಇದು ಜಾಸ್ತಿನೇ ಆಗ್ತಾ ಇದೆಯಲ್ಲ.
ಹೌದು ಸರ್, ಹೌದು ಸರ್.
ತುಂಬಾ ಇದೆ. ಆಲ್ ಆಫ್ ಯು ಸಿ kindly, all of you should be very careful. Don't go anywhere, so you should be very careful. Always use mask. Don't go to crowded areas, and wash your hands properly, and try to be warm with warm clothes. You should not get cold and cough. Always it should be, make it clear. So this particular thing, and of course now it is opened. Vaccination. I don't know whether in your place, how it is arranged in your respective hospitals. But vaccination is very, very important. Try to get yourself vaccinated.
Okay, sir.
Okay, all of you. Thank you all.
Thank you, sir. Thank you.
Thank you,
Thank you, sir.
Thanks. Bye.